Hello everyone, I welcome you all to today's session. I'm Shail, and in today's session we will see why DNA has thymine and RNA has uracil. As you all know, both DNA and RNA are different types of nucleic acids. DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. They both are made up of nucleotides. What are nucleotides? These are the complexes formed due to combination of sugars, nitrogenous base and phosphate molecules. The type of sugar which is seen in DNA and RNA is called as ribose. So, a nucleotide consists of a ribose sugar combined with nitrogenous base and a phosphate molecule. So this is one nucleotide. Many nucleotides join together to form a polynucleotide chain. So over here one more nucleotide will be added. See, phosphate, then one more sugar, nitrogenous base, phosphate. In this way, many nucleotides will be repeated to form a polynucleotide. Two polynucleotide chains are coiled around each other to form a DNA molecule, and a single polynucleotide forms RNA molecule. You all know these things. The nitrogenous bases are heterocyclic organic rings. These are heterocyclic organic compounds. What are heterocyclic organic compounds? They have carbon and some other atoms also in their main ring. See over here, nitrogen is also there in their main ring. So nitrogenous bases are the important part of the nucleotides. For there are four types of nitrogenous bases. In case of DNA, we have adenine, guanine, cytosine and thymine, whereas RNA has adenine, guanine, cytosine and uracil. In today's class, we will see why RNA has uracil and DNA has thymine. The nitrogenous bases can be grouped under two classes, as you all know, purines and pyrimidines. So purines and pyrimidines. If you have any doubt, you can revisit my lessons. My previous recorded sessions are there where I have talked about the basic structure of DNA. So purines and pyrimidines, these are the two basic types of nitrogenous bases. And these purines and pyrimidines can undergo non-enzymatic transformations. What are these? See, in case of DNA, continuously many types of errors or certain problem in their structure, certain transformations in their structure is taking place continuously. Let's suppose their nitrogenous bases, they undergo transformation. That is, let's suppose this cytosine undergoes certain loss. Let's suppose this amino group is lost and this cytosine forms something else. Next, during replication, let's suppose some wrong nitrogenous base is added. Let's suppose this thymine due to UV rays undergoes transformation and forms something else. So DNA is exposed or it is under pressure always. Its structure always undergoes some certain types of problems are seen in its structure always. These errors or problems are recognized and they are repaired by the help of various repair mechanisms. But if these errors or problems are remain unrecognized or unrepaired, they will lead to mutation. So what is mutation? Mutation is the permanent change or damage in the double-stranded structure of DNA, which is there because of the failure of the repair mechanism. That is, the repair mechanisms, there are various types like nucleotide excision repair, base excision repair. There are various types of repair mechanisms. So if the repair mechanisms, they do not recognize such problems or such errors with the structure of DNA, and if the problems they remain there, then that will lead to permanent problems, that is mutation. And mutations are carcinogenic in most cases. So among these various types of problems in the structure of DNA, there is a type of problem which is called as non-enzymatic transformation. What is this? The nucleotides in nucleic acids, that is, nucleotides are sugar, nitrogenous base and phosphate. In these nucleotides, these nitrogenous bases can undergo certain problems, certain changes, certain transformations without involvement of any kinds of enzymes and can form something else. Instead of normal nitrogenous bases, they can form something else. For example, over here, the cytosine over here, this amino group is lost from this without involvement of any enzyme, because of which it is called as non-enzymatic transformation. And this amino group is removed from here. It is replaced by this oxo group, and the structure we will get finally is called as uracil. When this amino group is lost and it is replaced by oxo group, then cytosine forms uracil. Let's compare the structure of cytosine and uracil. See over here, everything is same. The difference lies over here, the presence of this amino group and oxo group. So it is seen in DNA that cytosine normally undergoes deamination, can undergo deamination to form uracil. The structure what we will get is called as uracil. Similarly, adenine also undergoes deamination or removal of this exocyclic amino group and forms hypoxanthine, a structure called hypoxanthine. Guanine undergoes deamination to form a structure called as xanthine. Similarly, 5-methylcytosine undergoes deamination to form thymine. So these are the types of problems. Among various types of problems, this is a type of problem. In case of DNA, we will talk about this 5-methylcytosine later on. First, let me explain a few more things. So if these problems, they remain as such, that will lead to mutation. But mostly these problems are repaired by the help of various repair mechanisms. Okay. So in case of DNA, let's suppose there is one gene, and let me write that one: A T C G C G T A G C C G. Okay. Let's suppose this cytosine undergoes deamination and it forms uracil. Okay. So in place of cytosine we have uracil, and instead of C triple bond G, now we will have UG. Okay, that is U double bond G we will have. So CG is replaced with UG. Now what will happen? The repair mechanism will come. The type of repair mechanism to solve this is called as base excision repair. We will talk about this later on. The repair mechanism will come, and what will happen? It will see that instead of cytosine it is having uracil. It will remove this uracil. How? This uracil is attached in this way, phosphate. It will cut this uracil and remove this one. And in this way, what will happen? This uracil will be removed and it will be replaced with cytosine, and the problem will be solved. But this repair mechanism saw this uracil in case of cytosine and considered it as a problem because the uracil is normally not present in DNA. If the DNA would have uracil, how would this repair mechanism consider this a problem? See over here, as DNA is not having uracil at any place, DNA is having thymine. The uracil is a foreign thing, is a new thing for the repair mechanism. If the repair mechanism sees uracil at some place, it considers it as a problem because it is a foreign thing for the repair mechanism to be found in DNA. So it recognizes it very easily, removes it and repairs the problem, removes the problem. Because of this only, DNA has thymine. Let's suppose DNA has uracil instead of thymine. So let me write over here these two places. I will remove and replace with uracil. Now the uracils are present because in our example DNA is not having thymine, DNA is having uracil. Here cytosine has undergone deamination to form uracil. Now what will happen? The repair mechanism will not be able to detect this uracil because uracil is normally present. In that case, this uracil may undergo undetected. It may detect, but it may also undergo undetected or unrecognized and may lead to mutation later on. Okay. So because of this only, DNA has thymine. So this was the surface reason, the reason at its surface level. Now let's dig into the deeper levels. The first point being, the deamination of cytosine is the fastest as compared to adenine and guanine. The first thing is, the reason I have written over here, let me search. Okay, we will see it later on. The deamination of cytosines is 100-fold faster than adenine and guanine. If it is so fast, then then the cell has to take some solid steps. to solve this one. So the cell has taken the step ATCG. So the deamination of cytosine to uracil is 100 times faster than the deamination of adenine to hypoxanthine and guanine to xanthine. The next thing is, this adenine, when undergoes deamination, it forms hypoxanthine, which is not a type of nitrogenous base. It can be easily detected and removed. This guanine undergoes deamination to form xanthine, which is not a type of nitrogenous base. It can be easily detected and removed. So these two problems can be solved. So the cell is not taking any harsh steps to remove or to make this problem recognizable by the repair mechanism. But cytosine, when undergoes deamination, it forms a natural base. So cell has taken such strong step. Okay. So we are having a natural base uracil as a deaminated product of cytosine. The next thing. Then you would ask me, ma'am, thymine is also a natural base formed due to deamination of 5-methylcytosine. Let me answer that question. For that, first let us see what is 5-methylcytosine. 5-methylcytosine is a type of modified base found in DNA. DNA has modified bases as well along with normal bases. The one example of modified base is 5-methylcytosine. 4% of cytosines in our cells are methylated, or you can say 4% of cytosines can be methylated to form 5-methylcytosine, which is a type of modification. Why? Then we will see many things, much more things. Why they are methylated? This helps in regulation of gene expression. How? Let's suppose this is our DNA, and let's suppose this is one gene, this is another gene. These genes will undergo transcription to form mRNA. Okay. The genes are having promoters. As you all know, promoters are the part of DNA or gene where RNA polymerase binds and transcription takes place. So if let's suppose this gene will form mRNA, that means the promoter region will help the RNA polymerase to bind with it, whereas this gene will not form mRNA if the promoter region will not help in the binding of RNA polymerase. That means this promoter will be active, this promoter will be inactive. So if this promoter is inactive, that means this mRNA will not be formed. That means this gene will not be expressed. These promoter regions have many CG repetitions. GC. Okay. These repeated regions are called as CPG island. We will see everything slowly and steadily. Just concentrate. These are called as CPG island. These CPG islands are mostly seen in promoter regions, and these promoter regions, when have CPG islands, most of these C are methylated. That is, they are having methyl added to their 5th position to form 5-methylcytosine. And if this cytosine will be methylated in promoter region, then that gene will not be expressed. That means that promoter will not participate in transcription. So this modification of bases in case of promoter region, in case of DNA, helps the cell to regulate gene expression. This is an intentional modification of nitrogenous bases, and this modification has positive results. This is different from some error or some problem in structure of DNA. This is modification. So the modified nitrogenous base, example of modification in case of eukaryotes, is methylation on 5th position to form 5-methylcytosine. This 5-methylcytosine is modified, it is seen in CPG islands, and these CPG islands are seen in case of promoter regions of many genes. The meaning of CPG is C, phosphodiester bond, and G. That is, let's suppose this is our sugar, cytosine, phosphodiester bond, next sugar, guanine. So this P is, we are writing like in this way, and these 5-methylcytosines help in regulation of gene expression. I hope I have done this, I have explained this clearly. Now this 5-methylcytosine has a chance to get deaminated, and that forms thymine. But this thymine, even though it is a natural base, this case is very rare as 5-methylcytosines are found at very few places in DNA. And if the 5-methylcytosine is found at very few places, and even though they are found at very few places, the chances of them getting deaminated is very rare, and even though they get deaminated, they get easily detected by the repair mechanism. Therefore, the cell is not taking any harsh steps like the removal of uracil in this case as compared to these three cases. So these are the reasons why the DNA has thymine and RNA has uracil. So the cell has taken this strong step because of these reasons, the reasons which we have discussed already. So the normally 10 to the power 7 cytidine residues in 24 hours, that is 10 to the power 7 places where cytosine is present. Out of them, one may undergo deamination easily in 24 hours. That means there are 100 spontaneous events on an average in a mammalian cell, and this deamination seems very less at this step. But if this gets added upon, that is, they get accumulated, that will lead to a very big problem. Slowly and steadily, let's suppose CG we are having in DNA, CG, and this C if is replaced with U, then we will have UG, and slowly this G will also be removed and we will have UA. Slowly and slowly this CG will be removed from our DNA. This will lead to a very big problem as this deamination is the fastest as compared to the other types of deaminations. I hope I am clear now. Now, this recognition, if DNA normally contained uracil, recognition of uracil resulting from cytosine deamination would be more difficult, and unrepaired uracil will lead to permanent sequence changes as they were paired with adenines during replication. As I've already told, CG, C replaced with U, UG. Now slowly this G will be removed during replication and we will have UA. Now this will lead to a very big problem, and thus the thymine is one of the bases in DNA. Okay, over here I have talked about the repair mechanism, how uracil is removed. If anyone wants, can go through the slide. I will just tell few things. The enzyme which helps to remove that uracil, the deaminated form of cytosine, is called as DNA glycosylase, or you can say uracil DNA glycosylase. So uracil DNA glycosylase, what it does, it breaks down the N-glycosylic bond, that is, this is our sugar, uracil. This bond is broken and this uracil is removed during repair, and that is done by the help of uracil DNA glycosylase. And this glycosylase does not remove uracil from RNA or thymine from DNA. It is only sensitive towards the deaminated form of uracils. And if anyone wants, can go through this slide. And thus these things help the DNA to act as a better genetic material, as a stable genetic material. Because if, let's suppose, this is one generation of DNA, let's suppose CG, let's suppose this has deaminated to uracil, UG, let's suppose. In the next cycle, this will act as one template, this will act as another template. So we will have CG, this will act as parent strand. Then we will have UG, this will act as the second parent strand. Now replication will take place. What will happen? This GC, this will form a normal strand, but over here we will have we will have AC, the type of issue which will be formed generation after generation if that uracil undergoes if remains unrepaired. So the repair mechanism has to detect it as soon as possible and help the DNA to act as a better genetic material. I hope I have made this concept clear. If you have any doubt, you can ask me in the comment section. Very soon we will start with our live classes. First we will see the different topics of molecular biology. Then one by one we will cover many things. Thank you all. Thank you for watching. Please hit the like button, subscribe to the channel and share it with your friends. Thank you all. Thank you for watching.